So we revise, this is the third lesson for us to revise. We revise biological molecules and we revise transport in mammals. And we're revising this out of 11. So we have 5 more. Good. Fine. This is an important lesson, protein synthesis, with a lot of objectives. Now, what do I have here in front of me? This is the structure of what? The what? Oh, the nucleotide. Wake up, the nucleotide. We will first study the structure and then the making of proteins. The structure of the nucleotide, you will study 2 nucleotides, the RNA and the DNA nucleotide. It is made up of a pentose sugar, a 5-carbon sugar. The DNA nucleotide or the DNA sugar is the deoxyribose sugar. And Noor, fine, you have to know how to draw it. You don't have to know except what is present at carbon number 2 or 2 prime. That's it. Okay. So you have a 0 there, just like the picture here. 1, 2, 3, 4, 5. The carbon is going to move in a clockwise direction. This is carbon 1, 2, 3, 4, and carbon 5 is outside the ring. Hydroxymethyl group OH. The OH is going to be involved in the bond. That's why we have it like this. And then carbon number 2 is the only difference between the RNA and the DNA. The DNA 2 prime or carbon number 2, we have a hydrogen atom only. Okay? And in the RNA, Kareem, what do you expect we have in carbon number 2? We have a hydrogen atom at the 2 prime and the deoxyribose. In the RNA, OH. We have an OH instead of the H. That's it. Okay? That is easy to hydrolyze. That's why the RNA is not as stable as the DNA. That's it. So this is the difference. The RNA you have at carbon number 2 or the 2 prime an OH or a hydroxyl group instead of the hydrogen. That's it. Good. And the names of the bonds. Here you have a covalent bond that will bond you with a nitrogen base. In the DNA, it is adenine, thymine, uracil, and guanine, and they're held together by hydrogen bonds. AT bonds by 2 bonds, and the UG 3 hydrogen bonds. Okay. At CG23. And this is what? A covalent bond of which type? Who remembers? The phosphodiester bond. So this is the ester bond. There is the ester bond here and there's another one there, and together they make the phosphodiester bond. Okay? Now, bases are of 2 types, either purine and the pyrimidines. We said we will need to memorize the examples. The purine is the smaller name. So this is a double ring structure or a single ring structure. Double ring structure. And they include which? AG only. Okay. And the pyrimidine, this is the longer name or the bigger name. And this is the single ring structure. And they include the TUC, TUC. Okay, they always ask about the purines and the pyrimidines. Now, always one purine bonds with one pyrimidine. And this is to maintain what? To maintain what, Noor? The what? Tertiary structure? No. We're not talking about proteins at all. We're talking about DNA and RNA. DNA. The width of the DNA. The width of the DNA has to remain constant all through the length of the DNA. Because we have always one purine bonding with one pyrimidine. A double ring structure always bonding with a single ring structure. So this is going to maintain the width. Okay? And this is important because if this doesn't happen, let's say 2 purines or 2 pyrimidines, 2 double ring with 2 double ring, the width will increase. And this is going to be easily spotted and hopefully prevented so we can prevent mutation. Fine. Okay, Kenzie? So the purine and the pyrimidine, one is a double ring structure, one is a single ring structure. This maintains the width of the DNA all along the DNA and keeps the width constant. Okay, and then this is the importance of why one purine always bonds with one pyrimidine. Fine. This is the sugar here, the ribose and the deoxyribose. And by the way, these are both monosaccharides and they are considered reducing sugars. The only non-reducing sugar that you studied is the sucrose. Good. If you want to study the structure of the RNA and DNA, you can go ahead, but they don't usually ask you for the specific structure. They are exactly the same except for carbon number 2. Look at this. you have 1, carbon 2, carbon 3, carbon 4, carbon 5 is the hydroxy methyl group outside the ring, and the OH positions are important. If you go to carbon 1, the OH is up, carbon 3, OH is down. What is variable? Carbon 2. Okay, either you have OH down or H down. So if I draw it sideways here, the oxygen is here, carbon 1, 2, 3, 4, and CH2OH. Carbon 1, OH up, carbon 3, OH down. And it's carbon 2 that is variable. Either it is bonded to a hydrogen in DNA or hydroxyl in RNA. Clear? Okay? Because sometimes they ask you for the ratio of the oxygen and the carbon atoms. And the purine and the pyrimidine here, purine, double ring structure, and the pyrimidine, the bigger word, the single ring structure. We discussed that, so I want to make sure that we don't miss anything. The adenosine triphosphate, why do we study it? Because it is also a nucleotide. Okay? Here, again, we have a ribose sugar, the RNA sugar, the ribose sugar bonded to adenine base, only adenine base, no other base. And the ribose plus the adenine, those two, they are called together the what? Who remembers? The adenosine. The adenosine are the ribose plus the adenine. I highly recommend that you take notes. These will really help you on the night of the exam. And then adenosine, ATP. How many phosphates? How many phosphates? 3. Who said 8? 3. 1, 2, 3. They are bonded by high energy bonds. And every time one phosphate is released, it releases what? Energy. Every time a phosphate is broken, it releases energy. As every time we bond a phosphate, it will store energy. That is why it is the energy currency of the cell, or an energy molecule. Clear? Okay. Adenosine is ribose plus adenine. Adenosine is ribose plus adenine. Don't forget that. When you have only one phosphate group, it's called adenosine monophosphate, adenosine diphosphate with 2 phosphate groups, adenosine triphosphate with 3 phosphate groups. Easy? Easy up to now, very easy. All this, we're talking about the structure of the nucleotide, structure of the DNA and the RNA nucleotide and the ATP. Good? Now, the name of the bond, if you want to know its name, I never saw a question asking you about the name, but it is the phosphoanhydride bonds, and any bond is broken by a hydrolysis reaction. Most bonds are made by condensation reactions. Good, we said all that. Now, the structure. You have to know the structure. You have to know how to draw the structure. Okay, how do I draw the structure? Let's label it first. This is the phosphodiester bond. This is the phosphate group, and you have one ester bond here, one ester bond here. So this is called the phosphodiester bond. When you are asked to describe the phosphodiester bond, you can easily start by saying that it includes one phosphate group bonded to which carbon? Carbon number what, Adham? No, 5 here. This is 4, but you have the 5th carbon outside the ring. So the phosphate group, it is bonded by an ester bond to carbon number 5 to the nucleotide, to the same nucleotide it belongs to actually, and carbon number 3 in the next nucleotide. That is why it's called the phosphodiester bond. Each one of those is an ester bond, and then 2 of them plus the phosphate in the middle is the phosphodiester bond. Clear? Okay. This is the, here when you have a bond between another base, what is this bond called? Hydrogen bond. Okay, either 2 or 3 hydrogen bonds. Okay, this is what we need to know how to draw. We need to practice it because we will not draw 2 polynucleotide chains that are parallel. One of them is anti-parallel, and the 3' and the 5'. Very important. So how do I draw it? I can draw at least 2 nucleotides. This is one. Okay, this is another just below it. This is the oxygen number 1. And carbon 1, 2, 3, 4, 5. I don't need to number it. This is the base. And you have a phosphate group projecting out sideways. Good. I can get another color and just do this. So you here have 1 column of a polynucleotide chain. Now the opposing chain is usually the problem, Kenzie. Pay attention. I highly recommend that you do this trick. This is carbon number 1, here, carbon number 1. You will move horizontally, you will create an imaginary line so that you make it on the same level. You will move horizontally here, make a dot, another one here, make a dot, and you will draw the other polynucleotide chain inverted, okay? You have to practice doing that. And then you will go horizontally, go to that dot that you made, so that it is not out of step, and then you have to erase it and do it again. Good. And this is the oxygen, and you will move clockwise. This is carbon 1, 2, 3, 4, 5, the phosphate outside the ring, and the same case here. And then join the phosphate groups, and you will have an anti-parallel chain. The base will be at exactly the same level as the other base. And now you will have a full here diagram of the DNA molecule, the 2 polynucleotide strands, 1 parallel and 1 anti-parallel. Now, these 2 strands have different names. This strand with the phosphate group pointing upwards and carbon number 5 is the carbon available to bond to a next or another nucleotide. Is the 5' or the 3' Noor? 5', okay? So this is the 5' end. If you come to describe it, don't say it's the end with the phosphate projecting upwards. This is how it is described. It is the strand with carbon number 5 available to bond with the next nucleotide. Okay? And the other strand where the phosphate is pointing downwards, or you have a projecting OH group here, is which strand, Kenzie? The 3', pointing downwards. Okay? Anything, any enzyme, DNA polymerase, DNA, RNA, the RNA polymerase, any enzyme binds to which strand? To the 3' or the 5' strand? 3'. It binds to the 3' strand, so it will bind to this strand and build this strand. Okay, so any enzyme, put it in mind, rule of thumb, RNA polymerase, DNA polymerase, everything. Even the messenger RNA when it binds to the ribosome. Always the binding happens to the 3' end or to the 3' strand and building the new strand in the 5' to the 3' direction. Okay? The enzyme, here when we'll study the DNA replication, we have an enzyme that will build a new strand on a template strand, remember? So the enzyme is going to join one of the strands to build the other strand, to bind the bases according to complementary base pairing. It will bind to which strand? And to which end of the strand? The 3' end. Okay? For example, if you have in semi-conservative replication, first step in the semi-conservative replication is the unwinding and the unzipping, and then you end up with 2 template strands. You have one strand like this, okay, and the other strand anti-parallel. Yes? Fine. This is the 5' end, this is the 3' end, and here you have the 3' end to the 5' end. Now the enzyme will bind, if it binds to both strands, because it does bind to both strands in the semi-conservative replication, it will bind only to the 3' end. So in this case, it will bind here. I will bind here and build a new strand in this direction. Okay, in the other strand, I cannot bind here because this is the 5' and I'll move backwards and bind here. So I'll bind to the 3' end to build the new strand in the 5', 3' direction, in the opposite direction. Clear? So any binding of enzymes is always to the 3' end to build the new strand in the 5', 3' direction. So don't get confused. Yes, we will discuss it in the semi-conservative replication, don't worry, okay? But any binding of enzymes, they bind to the 3' end to build the new strand from the 5' to the 3' direction. Clear? Good. So first, you know how to draw it. You know how to draw this diagram. And we labeled it. And now we know which strand is 5', which strand is 3'. Clear? Any questions? Because you need to know the structure very well. We discussed all this. Everything we discussed. If you are asked to describe the structure of DNA, you have to give, or you don't have to, but it will be highly recommended that you draw this. You draw this, a small diagram on the side of the question within the writing lines, a small diagram for the 2 double strands, 2 DNA strands and the antiparallel. Draw it and describe it. You have two polynucleotide strands that are antiparallel, one 3', one 5', and talk about the bonds. And you have two bonds that you will mainly talk about, the phosphodiester bonds and the hydrogen bonds. And this is the sugar phosphate backbone. And here you have the complementary bases, and discuss the whole structure. But start with a diagram. This will really help you and it will really look good in the answer. Okay? Fine. So you will discuss this. The antiparallel strands, there are two. They're held, and you will talk about the bonds and the hydrogen bonds and the alternating deoxyribose sugars and the phosphate groups. So you can say a lot of things. The phosphodiester bond here, it is composed of a phosphate group bonded to a 5-carbon atom to one nucleotide and a 3-carbon atom of the other nucleotide of the sugar in the other nucleotide by covalent ester bonds. The phosphate group and the two covalent ester bonds make the phosphodiester bond. This bond is synthesized by the DNA polymerase enzyme. Clear? Okay. The DNA polymerase enzyme is not responsible for the hydrogen bonding, but it makes sure that the bases are complementary. So it proofreads. But the bonding, the hydrogen bond itself, is not one of the functions of the DNA polymerase enzyme, because the bonds, the hydrogen bond here, the bases bond due to their high affinity together at CG. Good. Now, we will not answer questions now, but now we explained this very well. This is here. Look at the phosphate. Where the phosphate is projecting up, this is the 5'. Phosphate projecting down, this is the 3'. Easy? Clear? It has been elaborated several times here. And the sugar phosphate backbone and the base, and then it is twisted into a double helix to take less space. Not only that, the DNA, the polynucleotide chain it coils, supercoiling over the histone proteins only in eukaryotes, again for the compact, more compact packing of the DNA so that it takes less space. So now you know the structure from A to Z. You know the structure of the monomer, you know the structure of the polymer, of the two polynucleotide strands, the antiparallel positioning, the 5', the 3', the bases, the coiling, the supercoiling over histone proteins, the polynucleotide strand coils over histone proteins. You know, you studied the DNA in detail. Clear? Okay. Now, this is too easy, the frequency of the bases, because you know the complementary base pairing, which is at CG. If this is 30%, T will also be 30% Noor. If C is 20%, G will be also 30%. And they get you a lot of questions in paper 1. You know this table where they get you the number of one base and they expect you to know the number of the opposing base. This is so easy. We'll discuss the semi-conservative replication. Okay? Now, remind me of the steps. What is happening here? This is step number 1, Farouk. Very good. Very good. Okay, so step number 1 is the, look at my hand, unwinding and unzipping. Unwinding and unzipping, because the DNA is coiled. Yes, we will unwind. It's like we will unwind in an anti-clockwise direction, and then we will unzip. The two strands will be split. The DNA helicase enzyme will split the two strands by breaking the hydrogen bonds between the two strands. The replication fork is the point of splitting, the point of separation between the two polynucleotide strands. Okay, Kareem? Good. Now, we will immediately start the replication. So you have another enzyme that will start the replication. Which enzyme? The DNA polymerase enzyme. These two strands are antiparallel. Okay, Noor, pay attention. So one strand is 3' to 5', and the other strand is 5' to 3'. Good. Now the DNA polymerase enzyme will bind to which end here? To the 3'. So the enzyme will bind here, and it will start building the new complementary strand in this direction towards the replication fork continuously in one go. So this strand will be called the leading strand. Okay, what does the DNA polymerase actually do? We have free activated DNA nucleotides. They will arrive from a nucleotide pool, and they will bind with the exposed bases by complementary base pairing at CG. Who is going to help in the making the DNA polymerase enzyme? It will create the phosphodiester bonds of the new complementary strand and its sugar phosphate backbone. Write all the keywords. and the bond, the phosphodiester bond, will be made by condensation reactions. Okay? Now, these two freely activated DNA nucleotides, we'll discuss them in a minute. Now, here the enzyme, can it bind Noor to this end? No, because this is which end? 3' or 5'? 5'. It cannot bind here. So it will go backwards and bind here and then move forward. Okay, as this is happening, a new part has been exposed. So the enzyme will go backwards again and then forwards. And the enzyme will go backwards again every time part of the DNA is exposed and then move forward. Okay, so the enzyme needs to go backwards and then forward. It needs to go move anti-parallel and then forward. So it will build this new strand on that side in sections or segments. These segments are called what? Okazaki. And then finally, we will fuse those segments together by creating also the phosphodiester bonds and the sugar phosphate backbone of those segments with the help of another enzyme, exactly, called the ligase enzyme. Good. When you are asked this classic question, you end by saying that you will have two identical DNA strands. Every DNA strand is made up of every DNA molecule is made up of one old strand and one new strand. And this old strand is known as the template. Okay, because both strands will be used as a template. Clear? So here you mentioned all the steps that are out of 5 marks. Yeah. Excuse me? It joins the Okazaki fragments together, the phosphodiester bond of the Okazaki fragment and the sugar phosphate backbone of the Okazaki fragment. Good. Now let's look at it in all steps. This is the unwinding and the unzipping, breaking the hydrogen bonds by the help of the DNA helicase. These are the free activated nucleotides. They will come and bind to the exposed bases according to the complementary base pairing. Here, the DNA polymerase enzyme is doing the work. It is doing a lot of things. Creating the phosphodiester bond, creating the sugar phosphate backbone. It is proofreading that the bases are complementary. Nice, okay, but it will bind to the 3' end to build the new complementary strand from the 5' to the 3' direction. I think you memorized it now. We will talk a little bit about the freely activated DNA nucleotides. Look at them here. Normally they have 1 phosphate, but how many phosphates do they have? 3. This makes them activated. What does the DNA polymerase do exactly? It breaks 2 of the bonds, as you can see here. And when this happens, this releases energy, and this energy is used in the making of the phosphodiester bond. So I want you to look closely at this moving GIF picture. See, those 2 phosphates will be broken in order to create the phosphodiester bond. And who will break and who will create the bond? The DNA polymerase. This is the condensation reaction that releases water. Okay? Any questions? Easy? Good. Again, unzipping, unwinding, unzipping. Helicase will break the hydrogen bonds. Here you have 2 ends, one 3', one 5'. The enzyme will bind here and move continuously. This will be the leading strand. But in the other strand, the 5' strand, I cannot bind. I have to move inwards. This is the 3. I have to move inwards and then backwards. And then I have to move here and then here. I have to go back and then forward, backwards and forwards. This is going to be the lagging strand because it's going to be built in sections. Okay, fine. Now you understand things very well. You have a problem expressing what you know on paper. This is what needs studying, to memorize what exactly you will write. Here, the notes will really help you. Write it with your own hands so that if you get this question, you know what to write and how to write it. This is the DNA. Why is it called semi-conservative replication? One old strand and one new strand. This is the DNA polymerase going backwards. Look at that. It will go back and then forwards. Backwards and then forwards. Move. It's not move. Backwards, see? And then forwards. And then go back again and then forward. Okay. The unzipping enzyme is the helicase. This is the primer, forget it. The DNA polymerase and the ligase enzyme, which glues the Okazaki fragments. So you need to mention at least 3 enzymes. Good? Okay. This question is very common. I think it's out of 5 marks. And they ask you about the bases and their names. Easy. Here we're still, again, when they ask you about the role of the DNA polymerase, you nearly write the same steps. Good. Any questions over DNA replication? And you have pictures here clarifying the activated nucleotides with the 3 phosphates, and then we remove 2, and this is going to make energy for the phosphodiester bond. Any questions over this part? Okay, and it's written for you so that you know what to write when you're asked this question. So we're done with the structure of the nucleotides, of the RNA, of the DNA, of the ATP, and the semi-conservative replication. Good? 1 minute break, and the break is finished. Okay, why do we have to keep one original strand? I mean, what is the importance of keeping of the semi-conservative replication? Excuse me? So it's not lost. So the parental information, so the original information is not lost. It's called genetic continuity, okay? I think leading and lagging strands are clear. This is a very clear picture. Done, done, done. We need all these enzymes, and we need, of course, energy. Then what? We talked about this enough. Oh yes, the experiment. The experiment is very, very, very important. Okay. We take a minute break or we move on? The problem is I will not be able to record. If I stop recording, it's your problem. The minute is finished. And I have hopes that we finish an exam and a half. Okay, this experiment. Now, why did they do this experiment? They wanted to do this experiment to confirm that the replication of DNA is semi-conservative replication. So what did they do? They got bacteria, E. coli, and then they put this bacteria in solution that contains the heavy nitrogen. The nitrogen is of two isotopes, the heavy and the light. It's not our problem now. So they put this bacteria in solution with heavy nitrogen for a very long time, so that the bacteria replicates until all its DNA contained the heavy nitrogen. And we started with this bacteria. So this is the beginning of the experiment, where we start with bacteria that contains only the heavy nitrogen, okay? We put this bacteria in another solution that contains now before we do that, so that they know the position of the heavy nitrogen, now they know that the bacteria contains the heavy nitrogen only. They will take the DNA from the bacteria and they will put it in a centrifuging machine that will spin, and then after the spinning is finished, they will now note down the level of the DNA. This is the level of the DNA that contains heavy nitrogen, or nitrogen 15. Okay? So this is the bottom of the test tube. So the bottom of the test tube represents the position of the heavy nitrogen. Good. What did they do next? They took this bacteria and they put it in another solution that contains only light nitrogen. And they left the bacteria only for 20 minutes. Why? To allow the bacteria to replicate once, because this bacteria replicates once every 20 minutes. What do they expect? They expected that the bacteria will replicate by the semi-conservative replication. So we originally had 2 polynucleotide strands with heavy nitrogen, 15 and 15. This is 15. If the semi-conservative replication took place, these 2 strands will split. This is 15 and this is 15. Every strand will make a complementary strand that is light, 14 and 14. If semi-conservative replication took place. Good. If semi-conservative replication took place. Noor, why do you look confused? Okay. And then they will leave it for 20 minutes for 1 round of replication. They said, if the DNA strand after centrifugation, after 20 minutes, settles in the middle, then all the DNA is a mix between the heavy and the light. All the DNA are considered hybrid, and this confirms the semi-conservative replication. And they actually found out after 20 minutes, they took the bacterial DNA, they put it in the centrifuge, spin, and then it settled in the middle of the test tube. No band was found at the base. This means that no DNA was 100% heavy. Remember, the position of the heavy was at the base. Now, when they put the bacterial DNA in a solution in nutrients broth that contains the light nitrogen, and they left it for 20 minutes, and then they spun it, I did not find any bands at the base. So here, no heavy DNA. The band now is in the middle. So now all the DNA became hybrid. All the DNA now became half heavy and half light isotope, or have light nitrogen. So this confirms the semi-conservative replication. What if the replication was not semi-conservative? There would be 2 bands, 1 heavy and 1 for the light DNA, but this did not happen. Clear? Okay, so this experiment confirmed that the replication is semi-conservative replication. Now, with every replication, we will use the light nitrogen to make the DNA, but the 2 original heavy strands will never go away. They will always be there. So what happens with every replication? I will have another band that is on top. This band will contain only the light nitrogen, and this band will be thicker and thicker and thicker, showing you that the ratio of the lighter nitrogen to heavy nitrogen is increasing. But there will always be a thin band here in the middle because the original DNA will never go away. So if you want to understand what happens, let's use numbers. I started with 15, 15. Every one of them represents one of the polynucleotide strands. And then these split and they make new strands, 14 and 14. So after the first round, the ratio will be 100% hybrid. And the ratio will be 1 to 1. 1 to 1 heavy and light. Are you all with me? Okay. And then this will split, and then this will split. This will split 15 and 14. And this will split 15 and 14. The 15 strand will make a 14 strand, and the 14 strand will make a 14 strand, because The only nitrogen available is the light nitrogen. The heavy nitrogen is not available anymore. The 15 strand will make a 14 strand, and the 14 strand will make another 14 strand. Now look at the ratio of heavy to light. The heavy is always here, 15 and 15, that's 2. And the light, 1, 2, 3, 4, 5, 6. 1, 2, 3. The heavy will always have a ratio of 1 because they will always be 2. They will never increase. They will always be the original 15 and 15. Clear? And with the second round of division, it will be 1 to 7. Okay? If you want to do it, split those two. This is going to give you a 14, 15, 14, and this will be 14 and 14. This will be 14, 14, 14, 14, 15, 14, 14, 14, 14, 14, 14, 14. Good? There will always be two 15s only. So they will be divided by a common factor. They will always end as one ratio. Ratio 1. The heavy is 1. Now the light, look at the 3 times 2 plus 1 will give you 7. 7 times 2 plus 1. This will be 1 to 15. Always the light will increase in ratio and the heavy will always be a ratio of 1. Clear? Okay? Now you need to understand it because they got a lot of questions here. This is before they mix it with the light nitrogen. This is the position of the heavy DNA at the bottom. All the DNA is heavy. I will mix it with the light nitrogen. It will now replicate by the semi-conservative replication. All the DNA will be hybrid. It will move and it will be in the middle. This is the position of the hybrid. Ratio 1 to 1. Okay? Now with time, what happens? The middle band never goes away because the heavy strands are always there. But the band above it always becomes thicker, as you can see, because the ratio of the lighter bands are increasing. 1 to 1, 1 to 3, 1 to 7, 1 to 15. Okay? Good. Fine. So we're done. Finished? Okay. The universal genetic code. The universal genetic code, this part is important. I was just looking at an exam that asks you about specifically about the universal genetic code and its characteristics. Where is it? This one? No. No, the exam that actually I want to solve with you. This one. We will revise it from here. We solved half of it online. No, not this one. Look here, yes. Here they ask you about the deletion and the types of mutation. Good. And then here, explain why the genetic code is described as universal. Explain why some mutations have no effect on the primary structure of the protein. Universal means what you tell me. We talked about the genetic code and its 3 characteristics. We said it is universal, it's degenerate, okay? And they're non-overlapping, okay? Now, described as universal, what will you say here? Yes, a genetic code is universal between us and other species and other organisms.
When each divides,
Yes,
always
code for the same amino acid, and every 3 bases are called the base triplet. On the DNA, they're called the base triplet. On the RNA, on the messenger RNA, they're usually called the codon. On the transfer RNA, they're called the anticodon. Okay, so here it's a direct question. So every base triplet codes for the same amino acid in different organisms. You have to mention that or clarify that the organisms are different. Explain how mutations have no effect on the primary structure. Yes, but why do we have this effect?
doesn't change the base sequence of the DNA.
Excuse me?
Doesn't change the base sequence of the DNA.
No, it does. The base sequence does change because one base is substituted with another base, but still the final protein is the same. Why? It's degenerate. Here you'll mention that it's degenerate. What is degenerate, Yassine?
Codons and code for The same amount.
The same amino acid, but here we're talking about the base triplets on the DNA. Different, yes, very good. You need to give the number. It's also in the answer. So different base triplets code for the same amino acid because we have 64 possibilities, because we have 4 to the power of 3, 64 possibilities of base triplets, but only 20 amino acids. Okay, clear? And this is the question. So this, they ask you, this is a recent exam. So going back to that part, we revised it. This is the genetic code. It's non-overlapping. We read them in threes. One base cannot be part of 2 codons. Good? So this is what it means by overlapping. Done? Done. Now the protein synthesis part. The part that someone came up and told me, I don't understand. We have transcription and translation. And then we have, after transcription, we have the post-transcriptional modification and the post-translational modification, which we actually took in another lesson. So what happens here? This is actually showing you both steps. We have part of the DNA, only part of the DNA will unwind. The part that we want to copy, because the DNA is made up of a lot of genes. I don't want all those genes. I want to copy part of the DNA, only this gene. Okay, so this part only will unwind. It will look like this. It will unwind. By the helicase enzyme, it will break the hydrogen bonds and exposing that part only. Okay, and then this part, only one strand will be used and one strand will be called the template. The other strand will not be the template. Then again, free activated RNA nucleotides will come from a nucleotide pool and bind with the exposed bases by complementary base pairing. What is the enzyme here that is responsible for the making of the messenger RNA? Not DNA polymerase, RNA polymerase. And again, it makes the phosphodiester bond and the sugar phosphate backbone of the new messenger RNA. This is transcription. Okay, this is transcription, copying part of the DNA that we want to synthesize as a protein. Okay, good. And you will mention that the hydrogen bonds will break, part of the DNA will be exposed, RNA polymerase, it will make the new messenger RNA according to complementary base pairing, and mention here AUCG, not ATCG. Discuss all the details. Good. Now, the messenger RNA is going to copy the whole gene. In the DNA, in eukaryotes, we have coding and non-coding base triplets. Yes, we have coding and non-coding parts. The coding and the non-coding parts, they have a name. The non-coding part, for what is it called? No, introns. Okay, so we have the introns and the exons. How do we remember them? The exons will exit. And these are the coding parts because these are the ones that we want to translate. So the messenger RNA will transcribe the whole length of the gene, and it will transcribe both the introns and the exons, the coding and the non-coding sequences you see. So this is the primary messenger RNA. Primary messenger RNA that contains the coding and the non-coding sections, the introns and the exons. What do we do to this primary messenger RNA before it goes out of the nucleus? We remove the what? We remove the, what did you say, Kanzi? Yes, we remove the introns and we fuse the exons together to create the mature messenger RNA. So this is the primary messenger RNA. Here we have the RNA, it will take a copy of the gene, and we will have the primary, messenger RNA that contains both the exons and the introns, We don't want the introns. So they will be cut, they will be spliced and removed, and the exons will fuse together. So we will only have a mature messenger RNA only made up of exons, splicing, introns removed, and the exons are fused together. This is the mature messenger RNA. This is the post-transcriptional modification. So the transcription, I'll take a copy of the DNA on a messenger RNA. The messenger RNA will copy the whole thing, both the introns and the exons. Good. After the copying is finished, I will remove the introns, fuse the exons. This step is called splicing. And then I will go out of the nuclear pore and fuse with the ribosome, and translation will start. Good? Okay, Noor? Good. Translation. So here it's going out. Translation. This is the transcription, the RNA, it will go out, it will give you a messenger RNA, and then it will attach to a ribosome, and we have translation. I want a moving picture. This is very nice. This is translation. Who will discuss it? Yes, Kanzi.
If it's an anticoder,
Yes.
binds to the messenger RNA,
Mm.
by writing bonds.
Mm.
I stopped coding is.
Very good. How many transfer RNA molecules can bind to the ribosome at any given time, Yasin? Hmm? Huh? How many transfer RNA molecules? Two. So the messenger RNA will bind to the ribosome. The ribosome will attach to the 3' end of the messenger RNA. And these three and every 3 bases on a messenger RNA are called a codon. I'll be quick. And this codon, yes, the transfer RNA, we'll discuss it now. And every codon is going to attract a transfer RNA molecule with a complementary codon known at one end. This is a complementary anticodon at one end, and the specific amino acid at the other end, called the amino acid binding site. So the transfer RNA will attach to the specific amino acid, bring it to the ribosome. Good. Let's yes, here. This is 1, 2, 3, for example. 1, 2, 3 here is the codon. This is the complementary anticodon. And on the other end, you have the amino acid binding site. It will bring the amino acid with it. And 2 will attach to the ribosome at any one time. And the 2 amino acids will bond by a peptide bond, which is also another condensation reaction. And then the first one will leave, and the third one will arrive, and the ribosome will keep on reading the messenger RNA until it reaches a stop Farouk codon. Okay? Stop codon. This is translation. What is, Adham, the post-translational modification? No. Introns are in the RNA. Wake up with me. These details are very important.
Okay.
Proofreading what? Proofreading, you will drive me crazy. And then what? Online, you don't attend. Offline, you're sleeping. What do I do? Here I have a cell. Okay, this is my nucleus. I have the DNA. I will take a copy of part of it, part only. Okay, I will unzip a copy. Okay, I'll copy. I'll create a messenger RNA. This messenger RNA will copy both the introns and the exons. I will remove the non-coding part, the intron, and I will fuse the exon. This is post-transcriptional modification. Nice. I will go out. I'll bind with the ribosome. The ribosome will read the messenger RNA. This is called translation. I'm going to read it in 3s. Every 3 bases on the messenger RNA is called the codon. And this codon is going to attract another molecule called the transfer RNA molecule. Here, the transfer RNA molecule that has a complementary anticodon and a specific amino acid. And another transfer RNA molecule with a complementary anticodon and a specific amino acid. No, pay attention. These 2 amino acids will come close together and bond. This first one will leave. A third transfer RNA molecule will come along, and we will create a polypeptide chain made up of a sequence of amino acids, the primary structure. Okay, Farouk? I'm done with translation. And I will stop until when I reach a stop codon. Good? Now, the post-translational modification, this is the primary structure. The modification of this primary structure of this polypeptide chain, Farouk, happens where in the cell? The Golgi body and the rough endoplasmic reticulum, they will modify, and you give examples. I will remove the first amino acid, the methionine. I don't want it, it's not functional. I will remove the final amino acid. Okay, I'm going to fold to create the secondary structure, fold some more to create the tertiary structure, and you know all the details, and fold some more and more to create the quaternary structure. I'll make it functional. I'll add a carbohydrate chain. It will be a glycoprotein. I will add a lipid. It will be a glycolipid. Okay, okay. These are the modifications. You need to give examples of modifications. Good. Now, this is a very important part of this, and this is very nice here, showing you now the transfer RNA. Here you have RNA bases that in some sections are held by hydrogen bonds, and it looks like a flower, clover. They call it clover flower. Anticodon and the amino acid binding site. And the last part of the lesson, is this clear or am I moving too fast? Any questions? In case they're asking you about the differences between DNA and RNA, the DNA is a double strand, RNA is a single strand, the DNA is larger, the RNA is smaller. Here we have a deoxyribose sugar, we have here a ribose sugar only. DNA, we have hydrogen bonds, RNA, we don't have hydrogen bonds, so the difference is very big. Okay, Noor? Any questions? the mutation. I highly advise you to study only this table. There are 2 pages before this table that explain if you want more explanation. Now, this is really important. What are the types of mutation? I have insertion, I have deletion, and I have the substitution, okay? Now, the insertion and the deletion, very obvious. I have here bases, and I will insert a foreign base or a duplicate a foreign base. What effect will this have on the final protein node and on the sequence of the bases? Yes, frame shift. It will have a knock-on effect or a frame shift. In other words, the sequence of the bases will change from the point of mutation onwards because I inserted a base, and we know that the base triplet or the universal code is non-overlapping. I'll read it in 3s. Clear? So I will have a different sequence of bases, different sequence of amino acids, different final protein. It's a drastic change. I did not finish. Don't count. Fine? And deletion. Tell me, Adham, what will deletion do? Again, I will remove one base, so the other bases will shift inwards. And this is also frame shift or a knock-on effect because the effect is going to happen from the point of mutation onwards. Everything else after that will be a complete mess. Different sequence of bases, different sequence of amino acids, different final protein, non-functional protein, or a different protein, which is going to be non-functional because it's not going to function the same way as the original protein. Okay? Now, substitution mutation. I will have a base, I will substitute it by another base. No frame shift, no knock-on effect, but it has other effects. Kareem, good morning. This substitution mutation, then what? By another base. What effect will this have, Farouk? It will change the base triplet, but this can change the amino acid, or maybe it will not change the amino acid because the base triplets are degenerate, or it might cause a premature stop early. Yes, this is called nonsense mutation, where it will stop early. Okay, it will make a very short polypeptide chain. Silent mutation, this is called silent mutation because of the base triplets that are degenerate. Or missense mutation, it will create another amino acid altogether. I will substitute one base by another base, and this is going to give me another amino acid altogether. So another primary structure, so another secondary structure, so another tertiary structure, and a final protein that is completely different. Clear? Okay? And you have a particular example that you need to study, which is the sickle cell anemia. We have the GAG original triplet. The A will be substituted by thymine. It will be GTG. The original amino acid was what? Glutamic acid, okay, which is water soluble, hydrophilic. And the mutant amino acid will be the valine. Need to memorize this example, which is non-soluble and hydrophobic. It will give me a sickle-shaped hemoglobin that is sticky and it blocks capillaries and blah, blah, blah. And this happens in the beta polypeptide chain, okay, the beta subunit, the beta globin in the hemoglobin, okay? Is everything understood? Because understanding is very important. You can write things in your own words. This is the good thing about Cambridge. Mutations, not necessarily one mutation will have an effect for all. Additional, when mutations add up, they can have a final effect. You took the cystic fibrosis in A2, صح? It's also a type of mutation. Okay, insertion. We discussed it, deletion. Here is a nice picture showing you when one is deleted, everything will be shifted sideways to the left. Substitution, this is the GAG. This is a nice picture here showing you the sickle cell. No, the A, not to G, to T. Nice? This is a nice one here. Silent, nonsense, missense. Silent, this is when there will be no effect. Nonsense, premature stop. Missense, a different amino acid altogether. Okay? We have 3 minutes. We solve an exam? Okay, now is this lesson understood? Any questions over this lesson?